Foodborne bacterial zoonoses. Foodborne bacterial zoonoses. Foodborne bacterial zoonoses. Definition. Human infection arises from the contamination of foodstuff originating from animal. इसमें पढ़ेंगे स्टेफाइलोकोकस ऑरियस, बैसिलस सीरस, क्लोस्ट्रीडियम प्रेफरेंस, क्लोस्ट्रीडियम बोटुलिनम, Escherichia coli, Salmonella species, Shigella species, Campylobacter species, Yersinia enterocolitica, Clostridium difficile, Vibrio cholera, Vibrio parahemolyticus, Listeria monocytogenes, Aeromonas, Hydrophila, Leishmaniasis. Foodborne viral zoonoses. Foodborne viral zoonoses. Rotaviruses, Norwalk virus, noroviruses, Norwalk-like virus, adenoviruses, astroviruses, calciviruses, parvoviruses. Foodborne parasitic zoonoses, Giardia, Lambelia, Entamoeba histolytica, Cryptosporidium, Parvum, Cyclospora, Catenalensis, Diphyllobothridium latum, Ascaris lumbricoides, Trichuris, Trichuria, Tinea sodium, Tinea saginata. Cause of emerging foodborne disease. Causes of emerging foodborne disease: demographics, consumer trend, change in food production. Demographics. An increasing world population places increasing pressure on global food production. Fueled by urbanization and higher income, there are change in the pattern of food consumption. Trend in food production. A trend towards global sourcing of raw material and processing in large centralized facilities and distribution of product over large geographical area using longer and more complex supply chain. Increasing number of localized catering and food preparation operation driven by consumer demands for out-of-home food consumption. Infection is caused by the growth of a pathogen in the intestine. Incubation time ranges from 12 hours to 2 weeks. Symptoms of infection generally include a fever. Intoxication due to ingestion of preformed bacterial toxin. Symptoms appear 1 to 48 hours after ingestion of the toxin. Fever is not usually a symptom of intoxication. Infection and intoxication cause diarrhea and dysentery, some gastroenteritis, usually treated with fluid and electrolyte replacement. Introduction. Salmonellosis, important foodborne disease of worldwide economic significance. An estimated 1.4 million cases of salmonellosis each year in USA. Between 1980 and 1994, reported cases of salmonellosis England and Wales 10,000 to over 30,000 per year. In India, endemic heavy economic losses every year. Salmonella, gram-negative, lactose-negative, rod-shaped family Enterobacteriaceae. Salmonella 2 species, Salmonella bongori and Salmonella enterica. Salmonella enterica 6 subtypes. Salmonella enterica subtype enterica, Salmi arizoni, Diarizoni, Horteni, indica. Host-range anthrophilic, S. typhi, S. paratyphi, zoophilic, S. gallinarum, S. pullarum, S. abortus equus and other. Zoonotic, S. typhimurium, S. enteritidis and other. Salmonellosis is a collectively description of a group of diseases caused by more than 2,500 seroviruses of the Genus Salmonella. Animal enteritis, septicemia, abortion, poultry, pulorum, fall, typhoid, paratyphoid, human typhoid, enteric fever, non-typhoidal infection, food poisoning, diarrhea and vomiting, gastroenteritis, hemorrhagic colitis, septicemia. 25410 words over 300 words within subspecies. First, CDC 360 words in human infection, 50% infection, typhimurium enteritis, Newport, 41.8% of veterinary infection, typhimurium Newport, CDC as typhimurium most common serovirus in USA, 65% isolated are as typhimurium among the total serotype. Increase in worldwide prevalence of salmonellosis animal. Interesting method in rearing, large-scale movement, inadequate control, commercial production of animal feed, improper disposal of animal waste product. Species are gram-negative, flagellated, facultative, anaerobic, basally characterized by OH and IV antigen. H antigen may occur in either or both of two forms called phase first and phase second. The organisms tend to change from one phase to other. The organism causing enteric fever possesses two major antigens, namely somatic antigen O and a flagellar antigen H, along with another surface antigen VI. Food as vehicle of Salmonella. Food as a vehicle of Salmonella. Egg, poultry, meat, pork, and other meat and products thereof are the common vehicle of salmonellosis to human. Improperly pasteurized fluid milk, ice cream, cheese, and other milk products, unpasteurized orange juice, uncooked tomatoes, raw alfalfa, sprout, etc. In India, food of animal origin have been found contaminated. Factor affecting growth and survival of salmonella in food. Factor affecting growth and survival of salmonella in food. The pH of optimum growth is around naturally 6.8 to 7.5, above 9 and below 4 being bactericidal. Minimum growth pH 4.5 within citric acid and 5.4 with acetic acid has been documented. At pH value more than 10, salmonella rapidly die. Aeration favor growth even at lower pH value. The temperature for optimum growth is 35 to 37 degrees Celsius, ranging between 5 degrees Celsius and 46 degrees Celsius. TMS also as 2 degrees has been reported for Salmonella typhimurium. Freezing arrested growth and may cause injury, but it is not bactericidal. Salmonella recovered from poultry and minced beef held for 41 at minus 18 degrees Celsius. Toxin, endotoxin, lipopolysaccharide, endotoxin complex, 3 component and outer polysaccharide coat, a middle portion, the R core, and inner lipid A coat. Released from bacterial cell wall as type P. Enterotoxin, 3 enterotoxin, protein associated with cell wall, produce fluid loss in infant mice, heat stable and rapid in action, heat labile and delayed response, both detected by vascular permeability of rabid skin. Cytotoxin act by inhibition of protein synthesis, damage to intestinal mucosa related to sugar toxin. Salmonella are destroyed at milk pasteurization temperature. Baked food are free of salmonella. Salt bind free water resulting in decreased water activity. AW below 0.94 is inhibitory. The growth of salmonella is generally inhibited in the presence of 3 to 5% NaCl. Brine above 9% is reported to be bactericidal. Prevention and control of foodborne salmonellosis. It requires multiple intervention along with coordinated control effort from farm level to the food procedure and the retail outlets, control of salmonellosis in animal, proper vaccination of animal and bird, early diagnosis of disease and immediate treatment and culling of diseased animal, avoidance of stressful condition to the animal, maintain of hygienic condition and pest control at the farm. Good manufacture practice GMP to improve food safety, hazard analysis, critical control point, HACCP approach to be followed to prevent manufacture fault, education of consumer and food handler on safe handling and cooking of food. Raw vegetable and green sprouts should be thrown washed and filled before use. Check at level of poultry meat processing. Checks at the level of poultry meat processing. Use of salmonella free feed. Incoming flow should be free from salmonella. Status can be known by microbial examination. Birds should come from houses containing dry litters of either soiling if there is minimal. Avoid cross contamination. One contaminated carcass has the potential to contaminate hundreds of following carcasses. Minimize the number of times the meat handled or has contact with anything else. Keep product as cool as possible at all stages of production. Vidal test. Vidal test. This test is used for detection of antibodies to ONH antigen of S type, group D, S paratype A, S paratype B. During infection with typhoid or paratyphoid bacilli, antibody against flagellar antigen of S type H, S paratype A AH, S paratype B BH, and somatic antigen of S type O usually become detectable in blood 6 days after onset of infection. Campylobacteriosis. Campylobacteriosis. General characters. Campylobacter species are gram-negative, slender, 0.2 to 0.5 micrometer wide, curved motile rod. Gram-negative curved rod with gomma S or gull wing shape. Daughter cell which remain jointed have a characteristic gull-winged appearance and long spiral formed by joint cell also occurs. Motile with a single polar flagellum. These micro-aerophilic organisms grow best on algae media in an atmosphere of increased CO2 and decreased oxygen tension. Many Campylobacter species grow on McConkie agar. They are non-fermentative and oxidase positive and have variable catalase reaction. No spore and no capsule. C. fetus subspecies fetus and C. fetus subspecies vulneralis are structurally usual as they possess a S-layer microcapsule. Campylobacter corkscrew-like motile or darting motile, vibrio rapid darting, shooting star motility, listeria tumbling and rotating. C. fetus, C. jejuni, coli, Larry, pylori. Usual habitat. Campylobacter species are found in the intestinal and genital tract of domestic animals and are widely distributed geographically. The principal disease conditions associated with infection are either intestinal, presenting as diarrhea, or genital, cause fertility or abortion. Campylobacter species were previously classified in the genus Vibrio, and the term vibriosis has been retained for some of the disease conditions which they cause. 3 species, namely C. fetus subspecies vulneralis, C. fetus species fetus, and C. jejuni subspecies jejuni are recognized pathogens of veterinary importance. A number of other species, some of which have been assigned to the genus Acrobacter, have been isolated from domestic animals and from humans. The pathogenicity of these species has not been clearly established. Usual habitat. Usual habitat. Many Campylobacter species are commensal in the intestinal tract of warm-blooded animals. Campylobacter jejuni and C. leri, formerly C. lerides, colonize the intestine of birds, which can result in fecal contamination of water courses and stored food. A number of Campylobacter species are excreted in the feces of pig. Campylobacter fetus subspecies vulneralis appear to be adapted principally to bovine preputial mucosa. Differentiation of Campylobacter species. Differentiation of Campylobacter species. Campylobacter species are strictly microaerophilic, requiring an atmosphere of 5 to 10% oxygen and 1 to 10% CO2 for growth. A selective enriched media such as spiro agar is usually used for primary isolation. Differentiation of isolation is based on colonial morphology and certain cultural biochemical and antibiotic susceptibility characteristics. Colonial morphology. Campylobacter fetus subspecies Vulneralis and C. fetus subspecies fetus have a small round smooth translucent colony with a divided appearance. Campylobacter jejuni produce a small flat gray colony with a spreading watery appearance. Colony of some Campylobacter species which may contaminated clinical specimens can be slightly pigmented. Because Campylobacter species do not fragment carbohydrate, other metabolic activities of these organisms must be used for identification. Toxin of C. jejuni. Toxin of C. jejuni. Enterotoxin. C. jejuni secrete a toxin with similar activity to cholera toxin and heat level toxin LT of E. coli. This toxin increases intracellular CAMP level and cause cytoskeletal rearrangements. Both toxin immunologically related bind to the same ganglioside GM1 on target cell surface. C. coli and C. leri produce toxin with cytotonic and cytotoxic activity. Cytotoxin, a heat and trypsin level protein of 70 kDa in size. 70 kDa cytotoxin neutralized by antibodies to Zika-like toxin. CA73 kDa protein inserted into target epithelial cell allowing subsequent invasion of the cell. A protein active on vero tissue culture cell. A cytolethal distending toxin with increasing intracellular CAMP followed by cell death. A protein having hemolytic activity, hemolysin. A protein that induces hepatitis in mice, hepatotoxin. Laboratory identification. Laboratory identification, specimen collection and processing. Fecal refrigerated and examined within few hours. Rectal swab in semi-solid transport medium. Blood draw for C. fetus. Care to avoid oxygen exposure. Selective isolation by filtration of stool specimen. Enrichment broth and selective media. Filtration pass through 0.45 micrometer filter. Microscopy. Gulving appearance in gram stain. Dirtying mortality in fresh stool rare done in clinical lab. Fecal leukocytes are commonly present. Identification. Growth at 25 and 37 or 45 to 43 degrees Celsius. Hippurate hydrolysis. C. jejuni is positive, susceptible to nalidixic acid and cephalothin. Botulism history. Botulism history. Germany, earliest recorded human outbreak. Organism isolated in 1895. Mortality rate of 5 to 50%. Long recovery period. Weaponized by several nations including the US, Japan and Soviet Union beginning in the 1930s. Iraq produced 19,000 liters of concentrated botulism toxin. Botulism toxin is the most poisonous substance known. Potent neurotoxin, highly lethality 5. Aerosolized gram could potentially kill 1 million people. Spore isolated fairly easily from soil could be released as an aerosol or as a contaminant in the food supply. Recovery needed long-term care, expensive. Botulism microscopy, botulism microbiology. Botulism microbiology, toxin produced by the bacterium Clostridium botulinum, anaerobic gram-positive rod-shaped bacteria. Bacteria are 0.5 to 2 mcm in width and 1.6 to 22 mcm in length. Created spores that can remain dormant for 30 years or more. Spore extremely resistant to environmental stressors such as heat and ultraviolet light. Clostridium botulinum, 7 type of botulism, A through G, based on the antigenic properties of the toxin produced. Toxin A, B, E, and F cause illness in human. Toxin C and D cause illness in bird and mammals. Toxin G pathogenesis unknown. Characteristic of pathological distinct C. botulinum associated with foodborne botulism. Characteristic proteolytic C. botulinum, non-proteolytic C. botulinum, neurotoxigenic C. botulinum. Neurotoxic format ABF, BEF, E. Minimum growth temperature 10 to 12, 30, 10 to 50. Maximum growth pH 4.6, 5, 4 to 5. Spore heating resistant more than 15, less than 0.1, less than 1 to 5. Food involved in botulism outbreak: home canned food, faulty processing, fermented marine product, dried fish, vacuum packed fish, vegetable-based food in Asia. Potential food problem: canned food, refrigerator processed food with long shelf life. Feature of the bacteria. Clostridium botulinum bacteria died at 212 Fahrenheit or 100 degrees Celsius. Clostridium botulinum spore die at 240 Fahrenheit 160 degrees Celsius. Botulinum toxicity natural at 185 Fahrenheit or 85 degrees Celsius. All the temperature must be maintained for at least 50 minutes and the heat must be consistent throughout the food, fluid and jar. Clostridium botulinum spore cannot has in strong acid solution of pH 4.6 or below. Some sources claim pH 4.7. Clostridium botulinum cannot grow, develop or multiply in food with a water content of less than 35%. Category of botulism. Foodborne botulism caused by eating food that contain botulism toxin. Intestinal botulism, infant and child and adult, caused by ingestion spore of the bacteria which germinate and produce toxin in the intestine. Wound botulism. Clostridium botulism spore germinate in the wound. Inhalation botulism. Aerosolized toxin is inhaled, does not occur naturally and may be indicative of bioterrorism. Botulism pathogenesis. Incubation period: foodborne 6 hours to 8 hours days, 6 hours to 8 days, 14 to 40 days. Inhalation 24 to 36 hours. Toxin enter bloodstream from mucosal surface or wound, bind to peripheral cholinergic nerve endings, inhibit release of acetylcholine, preventing muscle from contracting. Symmetrical descending paralysis occurs beginning with cranial nerve and progressive downwards. Can result from airway obstruction or paralysis of respiratory muscles. Secondary complication related to prolonged ventilatory support and intensive care. Botulism toxin mechanism. Botulism clinical presentation. Classic symptoms of botulism poisoning include blurred or double vision, muscle weakness, drooping eyelid, slurred speech, difficulty swallowing. Patient is febrile and alert. Infect with botulism with present with weak cry, poor feeding, constipation, poor muscle tone. Recent outbreak of foodborne botulism. Botulism infection control. Botulism infection control. Botulism cannot be transmitted person to person. Stringent precautions should be taken when caring for botulism patient. Botulism laboratory procedure. Toxin neutralization, mouse biopsy, serum, stool, gastric, aspirate, suspect foods, isolation of C. botulinum or toxin, feces, wound tissue, botex, fillers, Clostridium perfringens. Clostridium perfringens. Clostridium perfringens, gram-positive anaerobic spore-forming rod, widely distributed in the environment, frequently available in the animal and human intestine. Gas gangrene, potent toxin alpha and beta. Gas gangrene is characterized by several pain, edema, tenderness, and pallor, followed by discoloration and hemorrhagic bleeding and production of gas at the site of the wound. S2S. Pigwell consumption, large amount of for contaminated with C. perfringens type C. Trypsin is secreted in the gut and it can disrupt B toxin secreted by C. perfringens. Drum brand necrotic inflammatory gut disease. Perfringens poisoning is characterized by intestine, abdominal cramps, diarrhea which begin 8 to 20 hours after consumption of food contain large number of those perfringens bacteria capable for producing the food poisoning toxin that have been reported as a result of dehydration and other complications. Reported C. perfringens outbreak in USA. Prevalence of organism in food USA. Clostridium botulism, bacon 0.1%, liver sausage 2%, corn syrup 20%, honey 2%, Clostridium perfringens pork 0 to 39%, cooked pork 45%, beef 40 to 22%, chicken 0 to 44%, seafood 2.4%. Colibacillosis white score. Colibacillosis white score. Escherichia coli. Colibacillosis white scores. In 1885, Theodor Escherichia, a German pediatrician, first discovered this species in the feces of healthy individuals and called it Bacterium coli commune. Escherichia coli are common commensal inhabitant as friend of the gastrointestinal tract of animal and human. The bacteria mainly inhabited the lower intestinal tract of warm-blooded animals, including humans, and is repeatedly discharged into the environment through feces or wastewater effluent. The presence of E. coli in environment waters has long been considered as an indicator organism of recent fecal pollution. E. coli antigen structure: somatic, capsular, flagella, virulence factor, surface antigen toxin, or endotoxin activity can protect against the phagocytosis. Primary promote virulence, important in UTI. E. coli common hemolytic uremic syndrome HUS. Hemolytic uremic syndrome HUS, most common cause of acute renal failure in children. E. coli S70157, Zika-like toxin, varotoxin, abdominal pain, bloody diarrhea, fever, seizures, lethargy. Treatment: mainly supportive dialysis, no antibiotics, plasmapheresis, IV, IG. Nosocomial infection in hospitals and nursing homes may be caused by XPC strain, which may also be the cause of respiratory UTI or bacteremia in long-term hospitalized patients. Some animals, especially cattle, are thought to contribute disproportionately to transmission of infection and have been termed supershaded. Person-to-person transmission is possible among our crowded contact, child care, etc. E. coli 0157H7, E. coli 0157H7, E. coli 0157H7. It is one of the hundred of strains of the bacterium Escherichia E. coli. This strain produces a powerful toxin and can cause several illnesses. First recognized as a cause of an illness in 1982, most infections have come from the consumption of uncooked meat. WHO launched the Global Antimicrobial Resistance Surveillance System (GLAS). Biofilm. Pathotype: virulence factor. Symptoms in human: enterohemorrhagic E. coli HEHEC or Zika toxin producing STEC or virotoxin E. coli VTEC. Zika toxin STX first, STX 2, intimin BFPEAE, RBF 0157. Isolation and identification: diarrheal samples, suspected food material or water can be isolated by Enterobacteriaceae enrichment or McConkey broth, followed by selective media that is McConkey agar or eosin methylene blue EMB agar. The optimum incubation for bovine fishes to minimize overgrowth by other organisms in 6 hours at 37 degrees Celsius. For meat sample enrichment for 6 hours at 41 to 42 is used, and for water and dairy products 24 hours at 41 to 42. Biochemical test: cattle age positive, indole positive, methylene red positive, nitrate detected positive. Serological test: latex agglutination test, ELISA and monos resistant hemagglutination test. Molecular detection: real-time polymerase chain reaction, RT-PCR, multi-locus sequence typing, MLST, multiple locus variable number tandem repeat analysis, pulse field gel electrophoresis. Prevention. Prevention. Good hygiene, slaughtering practice reduce contamination of carcass by fishes, but do not guarantee the absence of STEC from produce. Maintenance of HACCP during production, processing and distribution of food. Basic good food hygiene practice recommended by WHO. 5 key to suffer food. Keep clean, separate raw and cooked. Cook thoroughly, keep food at safe temperature. Use safe water and raw material can prevent the transmission of pathogens responsible for many foodborne diseases and also protect against foodborne disease caused by STEC. Avoid raw milk, unpasteurized dairy products, and unpasteurized juice. Appropriate hygiene practice in farm and farm animal. Probiotic may be provided to cattle to reduce shedding of E. coli by competitive exclusion. Fembrile vaccine against ETEC may be used. Staphylococcus. Staphylococcus. Staphylococcus family Micrococci. Micrococci genus Staphylococcus derived from Greek staphyl branch of grapes include major human pathogen and skin commensal. Micrococcus skin commensal. Staphylococcus general characteristics: gram-positive spherical cell 0.5 to 1.5 micrometer in single pair and cluster, appear as bunch of grapes. Gram stain smear of Staphylococcus from colony. Staphylococcus general characteristics: non-motile, non-spore forming, non-capsulated catalase producing, oxidate negative, glucose fermenter, primary aerobic, some facultative anaerobic. Staphylococcus general characteristics: approximately 33 species, almost 50 species associated with human. Staphylococcus divided into coagulase positive and coagulase negative categories, inhibited by high bile salt concentration. Some are beta hemolytic. Colony morphology: buttery looking, creamy or white colored. Coagulase positive Staphylococci. Coagulase positive Staphylococci. Zoonoses or intermediate human pathogen. Hecus delphini salivary, animal associated species. Staphylococcus aureus, primary pathogen, habit anterior carrier, colonization, axilla, perineum, pharynx, produce superficial to systemic infection, mode of transmission, pus formation. Natural history of disease: usual site skin, nasopharynx, perineum, breach in mucosal barrier, can enter underlying tissue. Characteristic abscesses with pus, bacteria, liberated toxin. Disease due to direct effect of organism, local skin, deep abscesses, systemic infection, toxin mediated food poisoning, toxic shock syndrome, scalded skin syndrome. Virulence factor of S. aureus. Virulence factor of S. aureus. Pathogen factor: enzyme, catalase, control defect, coagulase, hyaluronidase, lipase, beta-lactamase, antibiotic resistant, toxin, enterotoxin, TSST, epidermolytic toxin. Skin lesions superficial. Skin lesions superficial: boil, stylage, furuncle, carbuncle, wound infection. Deep abscesses, direct or by blood, can be single or multiply. Example: breast abscesses, kidney, brain, osteomyelitis, species arthritis. Toxin mediated disease. Staphylococcus food poisoning due to producing of enterotoxin. Perform toxin heat stable, short incubation period, milk and milk product. toxin shock syndrome, high fever, diarrhea, shock, and erythematous skin rash, which disquamate mediated via toxic shock syndrome, toxin 10% mortality rate. Scalded skin syndrome, disease of young children mediated through minor staphylococcal infection by epidermolytic toxin producing strain, good prognosis. Virulence factor, extracellular enzyme, cytolytic toxin, alpha hemolysin lyses RBCS damages platelets, cause severe tissue damage. Beta hemolysin act on sphingomyelin in the plasma membrane of RBCs. Virulence factor, extracellular enzyme, hyaluronidase hydrolyzes hyaluronic acid in connective tissue, allowing spread of infection. Staphylococcus fibrinolysin, which allows spread of infection. Coagulase, virulence marker, lipase, allow colonization. Virulence factor, extracellular enzyme, beta lactamase or penicillin conifers resistant DNase degrade DNA protein A in cell wall. It binds to Fc part of IgG to block phagocytosis. Coagulase negative staphylococci habitat skin and mucous membrane, common human isolated as epidermidis saprophyticus. Coagulase negative Staphylococcus epidermidis. Virulence factor, slime, mode of infection, colonization of medical implant. Infection are acquired nosocomially. Serious infection among immunosuppressed patients or neonates may occur. Coagulase negative Staphylococcus saprophyticus habitat skin and mucosal membrane of the genitourinary tract. Common cause of urinary tract infection in young sexual active female. Laboratory diagnosis, direct smear examination, microscopic examination, gram-positive cocci, pair and cluster, numerous polymonuclear cell (PMNs). Laboratory diagnosis, culture characteristics, colony morphology, smooth, vitreous, white to yellow, creamy, grow well in 18 to 24 hours. S. oressus may produce hemolysis on blood agar. S. oressus. Identification test, catalase, principal test for enzyme catalase, drop H2O2 onto smear, bubbling, POS, step, no bubbling, streptococci. Identification test, coagulase test, cell-bound clumping factor converts fibrinogen to fibrin, which precipitate on cell causing agglutination. Extracellular enzyme free coagulase. 2 method, slide test, screen for clumping factor, tube test. Antimicrobial susceptibility, beta-lactam group of antibiotics, penicillin, fluoxacillin, ampicillin, amoxicillin. Beta-lactamase producers, treatment of choice, amoxiclavulinic acid or ampicillin, sulbactam combo or methicillin or oxacillin. For methicillin-resistant S. oressus MRSA, treatment of choice vancomycin. Thank you.